Hello everyone, my name is Michał Denkiewicz. I am a PhD student at University of Warsaw, also representing Warsaw University of Technology, perhaps even in different order. And I'd like to present to you today this very collaborative work about intrinsic linking of chromatin in human cells. So without further ado, we know that the human genome, about 2 meters long, is packed in a nucleus that is only a few micrometers wide and exhibits spatial organization due to this fact on multiple levels formed by interrelated structures such as chromatin territories, chromatin compartments, topological associating domains, and finally, as a basic building block of the structure at the bottom, of course, among other things, the CF-mediated chromatin loops. So while we can probe this structure via chromosome conformation capture technologies such as Hi-C, Chia-PET, Hi-chip, the principles of the organizations are still actively studied. Going to the very bottom, the chromatin loops are formed when a cohesin complex sliding on chromatin. Sorry, I was trying to show something with the pointer. When the cohesin complex sliding on the chromatin strand encounters two occupied CTCF binding sites with appropriate motif orientation. Thus a stable loop is formed, and this loop brings together remote locations on the genome. This can be around a few hundred kilobases, and this structural change has functional implications. It can either insulate or facilitate, for instance, contacts between genes and promoters and so on. So the loops are the basic building blocks, and what we consider here is the possibility that the loops themselves might be linked in 3D space like this. So this would not be, since this is a highly of course, random structure. It kind of moves freely in space. So this will not be really visible in chromatin conformation capture data. So far, loops linking like that and its relation to loops was described in the manner that loops could be a potential mechanism to localize such linking or even knotting in the way that the sliding cohesin complex would slide the linked region and to reduce its size and perhaps break it apart later. But here we investigate the possibility that actually the formations of these loops might actually provide some opportunities for linking. The way we do that is by studying graph representations of chromatin interactions. So we're not going to be looking at any matrices or as much build 3D models, but we're going to represent the contacts and the interactions or loops in a given region as a graph. Here, but in particular, we will base some work based on the Chia-PET assay targeting CTCF proteins. So we have those that captures the interactions that are mediated by CTCF. So we know that the CTCF is there, and we generally feel that these are confident loops. And we start with the interactions, and we construct the graphs in the following manner: that if we take the interactions, and if we have a loop, which is somehow represented in our data, we create an edge in the graph between two vertices. There are actually two vertices per loop anchor, but this is like a very technical detail. I don't want to get into that. And in this way, we create the graph. And one more thing that we do is we place so each vertex has a particular coordinates. We also arrange those vertices kind of on a straight line and add an edge between the consecutive vertices, thus forming the graph representation of the actual chromatin strand. And in this way, we obtain an actual graph representation of the loop-induced structure, which is a single connected graph. So why is it useful to study the structure in this way? Well, can't we just build like a 3D model using molecular dynamics? Well, since chromatin doesn't have a fixed structure, a given set of contacts can have a multitude of 3D representations. Some of them would be linked, and some of them would be not linked, and it is primarily the property of the actual starting initial seed structure of the molecular dynamics model that would determine the output. So this is not very robust. Doesn't tell us much. So we don't want to rely on that. Instead, we want to study the graph structures, which abstract from the particularities of 3D arrangement, and just look at the loops, and then just look at the general structure of the loops. When we exploit the fact that while a graph can have multitude of representations, there are some graphs that are called intrinsically linked graphs that necessitate linking. So if we have all those, if we have the structure present, then we know that somewhere in the structure there is a link. This is predicated on all of this contacts being in a cell at a given time. Nevertheless, it is necessary. And the basic theorem that we based on our research on is a theorem by Conway and Gordon from graph theory. So a graph is intrinsically linked, so it contains a link necessarily, if and only if it contains a clique of size 6. So this is a complete graph with 6 vertices as a minor. What does it mean? It contains as a minor. So here there is an example for a smaller graph of a K4. So the graph on the left, G, contains the graph on the right, H, as a minor. More precisely, G is a minor model of H. If the graph on the right can be obtained from G by contracting edges. So you can imagine that those yellow regions, yellow groups of nodes and edges, we can compact them into single nodes and form, and maybe remove some things, and form the final graph H. And if we can form the K6 like that, out of our chromatin contact graph, then we have, well, intrinsic linking. So this is like a high-view level overview of topology. There's a slight more detail into that. Not all minors are relevant for us because of the way we create and represent our graphs. For instance, we want to avoid a situation in which we have like a complete cycles of loops that are collapsed actually into a sort of packed structure like this, in which the cohesion itself would be somehow collapsed. We don't take this into consideration, so we filter those out. This is, however, it's not easy. It's exponential with respect to the number of edges in the graph. So we restrict ourselves to a smaller subset called of minors, called linear minors, in which we consider only those regions linearly present on the strand, which are consecutive. I'm just going to very quickly say the general gist of the algorithm. We take advantage of the fact that we can split. We use something called path decomposition of the graph, which takes advantage of the fact that these graphs are kind of have a linear structure, and using this path decomposition, we can then construct those minors on the fly. By dynamic programming, it is still an exponential algorithm, but it is not exponential with respect to number of nodes of edges, but on the certain width parameter called path width that is and that is devised from this path decomposition. And for in practice, we encountered this parameter to be like 9 through 24, maybe slightly more, so it's manageable. So the particular data that we used to run this algorithm on was ChayaPET targeting CTCF, like I mentioned, and we used long-reach ChayaPET for the human lymphoblastoid cell line, and the in situ ChayaPET for sudden cell lines for the nucleome. The long-reach ChayaPET was provided courtesy of the Jackson laboratory. So we focused primarily on GM 12878, and there was also the human embryonic stem cell line, foreskin fibroblast cells, and human induced pluripotent stem cells. And also to keep the thing manageable, we subdivide the data into chromatin contact domains. It's a concept similar to the concept of topologically associating domain, but it's defined on the basis of ChayaPET. These are smaller regions that are contained mainly within region contacts. So this seems to be a good way to divide it. We found a large number of regions that are candidates for linking with this linear algorithm, which I can remind you that it's also searches only for only specific class of minors, so it's not everything. So potentially the full algorithm would be would detect more. So they like thousands of minors occupying like 16% of all CCDs that we of all regions that we detected. For in situ ChayaPET, it's slightly less, and of course this highly depends on the particularities of the experiment from which the data came from. So we got much less for especially for human embryonic stem cell line. We have some ideas why that might happen. In general, we can't exactly establish some sort of correspondence or pattern. So I'm just going to give you an example of a linked region that we found in the long-read data. So I selected this region just because it's quite compact. So we have in the bottom right we have the graph that highlights that it represents the linked region. The whole CCD is modeled using Spring Model, our in-house chromatin modeling tool. The whole CCD is modeled here, and here is the zoom of the actual linked region. You can see the linking here. Of course, again, not all those loops have to be here, but at least two of them have to. And if you look at it closely, you can see that this structure cannot be disentangled. In general, it's hard to obtain such a linked structure in molecular dynamics just by chance. And so this is required here. So what we did also was to try to characterize those linked regions. So are there any different in terms of structure? So we studied basic graph properties of these regions. So these regions are seem to be a little bit larger, but slightly less dense. So this can be together construed as the fact that since density falls off with edges in a square fashion, then we can say that density is not greater in those regions, but those regions are generally larger. They also have a higher tendency to contain compartment boundaries, some boundaries between A and B compartments. It's not like definite, but there's this variability in proportion between non-linked CCDs and linked CCDs. To further characterize those regions, we wanted to have a more in-depth knowledge about the structure of those regions. We use for this the centrality measures which are known and widely used tools in network science that can capture in a certain framework that can capture the general structure of a graph. So we can distinguish between graphs like this and graphs like these. And what we see is that those regions have both low closeness and betweenness centralities. So those are two main centralities that are most prevalent in the literature. So we understand this as having those graphs having a number of redundant connections and subparts of these regions are clumped together. So this is concordant with what we see in the simulations here. So to conclude this, we provide an algorithm that finds potential links, well provided on the underlying CHAPET data is accurate. And our method found multiple regions with noticeably different network characteristics, tending to contain compartment boundaries more often. And we propose that this actually can the chromatin loops can not only remove linking but sometimes cause linking. And the implications would be that the presence of a stable link would impact the overall structure, because linking prevents parts of the chromatin from moving apart in 3D space on a larger scale. So it might have in the future so some functional implications. So at this point, I'd like to thank you for your attention. I'd like to acknowledge all my coworkers, especially Maciej Borodzik, Dariusz Plewsztyński, Kamila Winnicka, who initiated this project and devised this general idea. Also, Michał and Marcin Pilipczuk, and Krzysztof Spański, who provided the basis, the bulk of the implementation, and also Kastav, who helped me with some analysis, and of course Professor Plewsztyński for supervision, and Ijan Ruan from the Jackson Laboratory for providing the necessary data. So again, thank you, and I'm open for questions. I also have a poster about this now, so if you have something more, some longer questions, we can talk later. Sorry.
Thank you very much. Are there any questions?
Yes, actually, I have a question. So I don't
Speak closer to the mic.
So I don't know much about the chromatin and like come together, but from your graph, it looks like it's very tangled. What prevents chromatin from tangling like permanently and being unable to express what? Or would it be able to express even with such a tangled chromatin?
So to summarize. Yeah, so when I will go back to the beginning. So the idea that was in the previous research was that when we have a loop, it can slide and like restrict this tangled region to a specific area, and then it would be simply this topoisomerase will simply cut this region open, and this tension will be relieved, and this will be removed. In fact, this is also, I think, required for formation of the links that we see as well. Yeah, because somehow we need to get those loops attached.
So chromatin topoisomerase is involved also in the chromatin clumping, I guess. So it may be like making breaks and everything. It can form this structure or loosen them, right?
In the interest of time, I would ask you to get together at lunch break.